The overall goal of this procedure is to profile microRNAs in cerebrospinal fluids by quantitative PCR and to analyze the data using GenX professional software. This is accomplished by first isolating total RNA from cerebrospinal fluid samples. The second step is to perform reverse transcription in order to transcribe the RNAs into cDNAs. Next, the cDNAs are mixed with a cyber-green master mix, loaded into 384 well plates containing primers for 742 unique sequences, and amplified using real-time PCR. The final step is to analyze the data acquired from the real-time amplification. Ultimately, the results can be expressed as a fold change increase or decrease over a control, and visualized in various graphic representations.
So the main advantage of our technique over existing methods like chip arrays is that our method is highly sensitive and requires minimal amount of RNAs. Generally, individuals new to this method will struggle because of the high amount of data generated and the use of the software for data analysis.
First, spray the working area and pipettes with RNase ZAP. To begin cDNA synthesis, dilute the template RNA samples to a concentration of 5 nanograms per microliter with nuclease-free water. Next, gently place the 5x reaction buffer on ice to thaw. Then, add 40 microliters of nuclease-free water to the RNA spike-in to resuspend the reagent. Vortex the tube and leave it on ice for 15 to 20 minutes.